Good day, ladies and gentlemen. I welcome you to our second lecture titled Experimental Characterization in Immunology and Immunobiology. This lecture will focus more on molecular characterization, chemical characterization, and structural determination of immunological tissues, organs, and cells, most especially in the area of molecular interaction, the chemical kinetics, rate of binding between antigen and antibody, and also the characterization of different types of antigen from different pathogens or etiological agent. It is also major the techniques for the structural determination of these immune cells and organs. Therefore, we are going to discuss much on surface plasmon resonance, experimental characterization techniques in immunology and immunobiology will determine the experimental methods, laws employed, and techniques used, mechanism and the processes in vitro and in vivo that we are using in understanding how immune cells are interacting in the human body. So we are going to discuss surface plasmon resonance and its relationship with immunology, X-ray crystallography and cryo-electron microscopy, also its relation with immunology, and mass spectrophotometry, propylene, and God willing electrophoresis analysis, which I know some of you are more conversant with it. The application of surface plasmon resonance in immunology aimed at determining the rate of kinetics, as we earlier explained in the first topic, the rate at which antigen and antibody are binding together when there is a conformation between the paratoph and the epitope. Earlier explained the affinity is just a rate of binding between one antigen and one antibody, where the avidity is the general overall binding strength between a complex of antigen and antibody, which we explained that the valency matters. The concentration of the antigen and antibody determines the strength of the bonding between the antigen and antibody complex. So the surface plasmon is just a technique which we use in vitro to detect the rate of binding between this antigen and antibody. So it detects changes in the refractive index near the sensor on the surface when the molecules are binding, because if let's say you pass a light through a dense concentration of antigen, the refractive index will be smaller. But if there is a complex between antigen and antibody on the surface of, say, any substances you pass a light, that means the density changes, so there are changes in the diffractive index. So the baseline behind this is that we are not only measuring the strength of binding, but also the rate of how much they associate together and how much they dissociate together. At the end of this experiment, there is a sensor which detects the changes in the rate of dissociation and association. We are using this value to calculate the equilibrium dissociation constant by dividing the rate of dissociation by the rate of association. So the smaller the equilibrium dissociation constant, the higher the affinity between the antigen and antibody, and the larger the equilibrium dissociation constant, the lower the affinity between the antigen and antibody. So this is a yardstick we use in calculating the rate of association and dissociation between antigen and antibody reaction. During the procedure, first of all, the antibody will be immobilized on the surface of a gold sensor, the equipment. Then antigen will be plotted over the surface. Then the binding, the rate of the association and dissociation will be measured by passing a ray of light through the complex, either during the association and dissociation. At that very moment, a sensor will collect the reflected light so that it can measure the strength at which the antigen and antibody binded and the rate at which the antigen and antibody dissociated at in real time. So a more clear depiction here, you can see the procedure pass through 4 stages. There is immobilization, when you add your antibody, then you mediate your antibody on the surface of the gold chip. Secondly, you associate the antibody and the antigen by flooding them with the specific antigen. Then by passing the light through, you can measure the rate of association between the antigen and the antibody. This is happening in real time. Then during dissociation, you are going to add a buffer, which this buffer, by concentration, it can alter the conformation between the paratoph and epitope, so the antigen and the antibody will dissociate. By this dissociation, at real time, again, you are measuring the rate of diffraction of light. Then the sensor will collect the information all together and plot a graph of association and dissociation, which can be clearly calculated based on the value collected using KD is equals to K dissociation and K association. So the smaller the rate of the association, the higher the affinity, and the larger the rate of dissociation, the lower the affinity. So the surface plasmon resonance has a vast application in immunology. It's cut across, first of all, measuring antibody antigen interaction, so that is the strength. Then there is epitope mapping, the nature and the morphology of the epitope on the surface of the antigen, because you know we have so many variable antigen across so many microorganisms, so they have different epitope binding site, so we can map them using surface resonance by how much light it diffracted and how the sensor collected it via 2D, then reform it into 3D to form a structural conformation. And there is we can also measure the screening the morphology of the antibody to see how they are conforming with the antigen. We can also use it as a diagnostic by a sensor to detect the presence of your antigen in a blood sample. As well, in vaccine development, we can use in screening the antibodies to see how their variable region is formed and if it will be effective in binding with the specific test antigen. Another immunological technique is the X-ray crystallography. This is operating based on the principle of diffraction of light, and an immune sample will be cryogenated using a cryogen, which can be, there are a lot of cryogens. Then a ray of light will be passed through the cryogenic immune cell. Detector plate will collect it, then the sensor will reform it into a 3D structure. To analyze this technically, we use mathematical formula applying the law of Briggs or Bragg's law, which means n times lambda is equal to 2d sine theta, where n stands for order of reflection of the light, lambda the wavelength strength, the strength of the X-ray passed through the cryogenic immune sample, indeed the distance between the crystal planes as displayed on the detector plates, whereas the theta stands for the angle of incidence. We use this formula in calculating the structure and the rate of the 3D as replicated on the sensor. From this depiction, you see that if a ray of light is passed through the protein crystal or immune crystal, there will be diffraction of light. This diffracted light will be collected by a detector sensor, and the detector sensor will 2D map of the immune cell within the crystal. Then it will later be reformed into 3D molecular structure. Technically, that's how we detect microscopic structures, so that we can know their morphology, their shape, their symmetry. In the case of a antigen-antibody reaction, the structure of the antigens, the sites of the epitope and the paratoph. So the procedure starts by crystallizing the immune cells, be it either the protein or the antigen-antibody complex, using a cryogen. So when it is crystallized in such a way that it is immobilized, it cannot move, but the structure are intact without being breaking. You know, antigen and antibody, they are very flexible. because for the case of the antibody, it is linked by a disulfide bond, which is very flexible. So that's why cryogenation is a very delicate procedure. So after the cryogenation, then the X-ray light will be passed via the cryogenate sample. Then it will be detected by a plate. The crystallography is used in detecting molecular structure and arrangement of antigen and antibody binding site. Also the structure of immunoglobulin and the immune receptor interaction, that is between the paratope and epitope. So it's very significant in mapping out the reaction between immune cells. The principle of operation between crystallography and cryo-electronic microscopy is technically the same. However, what differentiates them is that in the case of the cryo-electronic microscopy, the immune samples are processed using liquid ethane, liquid nitrogen. These are technically cryogenated. The biological samples or immune samples immediately in such way that it's not developing like an ice due to short time it takes to crystallize. In this case, the electronic beam passes light through the cryogenic sample, scattered at the diffracted ray is collected by a single particle analyzer, which technically receive the diffracted light in the form of 2D. Then a computer algorithm change it to a 3D image. The structure will be manifested and the reaction can be seen in real time. This depiction, you can see that electronic transmission microscope. Then under the visualizer, a cryogenic sample will be placed from there. Then a beam of light will pass through the transmission microscope. It will be detected on a plate in the form of 2D. Then the algorithm will change it into 3D. So you can see the Y shape of antibody. So it is just a transmission from a cryogenic sample into 2D. Algorithm will rise it off into 3D for clear visualization. So in the procedure, the sample will be preferred. You know, preparation of sample, it can be either antigen or antibody, it can be immune cell, it can be immune tissue, it can be an immune complex. However, before we collect it from either the blood circulation, first of all, we have to homogenize it, collect the fragment, then will be separated either using electrophoresis or liquid chromatography. Plunge into prison. Vitrification. It's not just direct prison, but it is an immediate prison where a tiny drop of the sample will be placed on a carbon grid. Then it will be bloated to make sure that there is a thin film formation. Then directly it will be plunged into either liquid nitrogen or liquid ethane at approximately minus 180 degrees Celsius. As the sample is set on the transmission electron microscope, then the rate of light will be passed through the cryogenated sample. It will be detected by a metal plate with a sensor. This will be a collection of thousands or hundreds of diffracted isolated light, and it will be collected in the form of 2D. For the image to be processed, there are a lot of other softwares that can be used. This includes Relion or Cryospark, which align the image together using selected diffracted light. Technically, it will form a 2D image. Then the 2D image will be reformed again using an algorithm that will reconstruct the image collected via diffracted light. Put atomic model of the amino acid into a 3D density map to determine the exact point of interaction between the antigen and the antibody. So technically, they are similar with the crystallography, but the process for the preparation of the sample and the softwares used in constructing the 3D structure varies across the equipment used for cryo-electron microscopy and crystallography. This is a pictorial representation of the procedure. As you have seen, the sample will be prepared. After it is prepared by either liquid chromatography or electrophoresis, then it is separated. It will be applied on a sheen or grid. Then it will be blotted to make sure that a thin film is formed. The vitrification is just the use of the liquid nitrogen or liquid ethane to reduce the temperature to minus 180 degrees Celsius, which instantly will form a cryogenic sample. And the microscope will pass the beam of light to be collected in thousands, forming a 2D structure. Then the imaging processing software will create a 3D structure. Now, due to clarity and the standard and the sophistication of the cryo-electron microscopy, which is just a transmission electron microscopy, it has a vast application. First of all, it's a method that you can detect the interaction between the reactivity of antigen and antibody in real time. So that's why it's highly sophisticated, and it is also a way where you can notice the mechanism of the complement system. You can see how the complement proteins are attaching with antigen and how they can employ membrane attack complex to make sure that they create a hole or make sure that they perforated a bacterial cell wall to destroy it entirely. As well, you can see how you can visualize a massive T cell receptor and cluster of differentiation tree complex as they form on the surface of the immune cell, and how they attract by sending a signal to B cell so that the pathogen will be recognized. Using this method as well, you can notice in real time how inflammatory cells and molecules they are gathering in a place where there is the availability of antigen, or in a wound immediately after there is a traumatic implantation of an antigen on the surface of an individual. So that's why it is state of the art, and we call it a gold standard. Another method for detecting immunological parameters and mechanism is a mass spectrophotometry profiling. This is derived from spectrophotometry, as you know in chemistry. Then the process will continue as I've earlier explained. Through the acceleration, the power of the magnetism will differentiate them or separate them by charges because they are already ionized, and also by time of flight based on their density or mass. So the higher the density, the slower they will be, and the lower the density, the mass, the speeder they will be. So the time they arrive at the detector matters. It can use to detect the chemical composition of the analyte. That's a vast application. One of the applications is immunopeptidomics, identifying the exact fragment of the antigen, which can be either fragment of viruses, fragment of tumor cell, fragment of other organism on the surface of the either T cell. If it is T cell, on the surface of the major histocompatibility complex. If it is B cell, on the surface of B cell. It also applicable in the biomarker discovery, where the protein profile of a healthy person individual will be compared with someone with autoimmune disease to find early warning proteins as a diagnostic technique. They can also measure the concentration of cytokines and interleukins. There are also metabolic programming, where scientists are studying how immune cells change their fuel sources. As fuel sources, the presence of ATP on the surface of the B cells after they have been sorted out, when they will be switched from a resting state to an active state. So all these are very effective in a scientific discovery and understanding the chemical dynamics, chemical kinetics, and chemical structure of these immune cells. So thank you for listening once again. You see that there is another link down there is for your second MCQ and attendance, but you are advised that to make sure that you watch the lecture. If necessary, I'll provide a link for a question. If you have a question, you can drop it down there, I think before the MCQ or after the MCQ, but make sure that you attend to the MCQ because it is attendance, and I may use it as an assessment. So thank you. I will respond to your question if there are, and we'll see for the next class. Thank you.